This is Bio 182, Lecture 1.2, Genetics Review. Heredity is the passing on of traits from parents to their offspring. Phenotypic traits are characteristics of an organism that can be observed, such as blue, brown, or hazel eye color. The study of heredity in biology is called genetics. Traits are inherited by passing on pieces of DNA called genes. A gene is a piece of DNA that codes for a protein. Genes have different versions called alleles. Alleles make proteins that have different shapes, which result in different properties for that gene and for that protein. Those properties cause differences in the phenotype or the physical manifestation of that gene. Humans and many other organisms are diploid, which means that they have two copies of each gene. One is inherited from mom and one is inherited from dad. In a diploid organism, homozygous means that the individual has two copies of the same allele for the same gene, such as two tall alleles for height or two alleles for blue eyes. Heterozygous means that the individual has two different alleles for the same gene, such as one wrinkled and one smooth allele for P shape. Alleles and genes have different versions, and they are either recessive or dominant alleles. A recessive allele is not expressed in the presence of a dominant allele. It is only expressed if it is homozygous recessive, meaning that the individual has two copies of the same recessive allele. If a pea plant is heterozygous, would it be wrinkled or smooth? If the wrinkled allele is recessive, the pea will be smooth. If the wrinkled allele is dominant, the pea will be dominant. A dominant allele is the one that is expressed if an organism is heterozygous or homozygous dominant. A recessive allele is not expressed unless the organism is homozygous recessive, meaning that it has two recessive alleles. There is a difference between the genotype and the phenotype for an organism. The genotype is the sorts of alleles that an organism has. That is determined at the molecular level, it's in their DNA. A phenotype is what the organism's trait looks like, such as round seeds. Individuals all have both genotypes and phenotypes. In this case, when we're talking about the shape of peas, genotype can either be homozygous dominant, which is big A, heterozygous, which is big A, or homozygous recessive, which is little a. And the phenotype is determined by the genotype. So if the individual is homozygous dominant or heterozygous, it's going to have round shape. And if it's homozygous recessive, it will have wrinkled shape. A Punnett square is a diagram that we use to predict the genotypes of a particular cross or a breeding experiment. In a Punnett square, we take the two alleles for each parent and put them on the top or the side of the square, and then determine the probability of the genotypes and therefore the phenotypes of their offspring. When we construct a Punnett square, it does not matter if the female or the male is on either side. They can be interchanged as long as we have the full genotype for that gene, both alleles. We can then use that information to determine the genotype and then the phenotype of their offspring. In genetics, we refer to traits that are inherited through one gene as simple Mendelian traits, and they are inherited through simple Mendelian inheritance. This term of Mendelian inheritance comes from a monk. His name was Gregor Mendel, and he used pea plants to do experiments between 1856 and 1863 and was able to determine many rules of heredity. And we now refer to those rules as the laws of Mendelian inheritance. In his experiments, he used crosses between pea plants with different colored flowers to make inferences about inheritance. So he used either true breeding violet flowered pea plants or white flowered pea plants and would breed them with each other. True breeding just means that they're homozygous. Neither one would be a heterozygote. So in his experiments, he took homozygous violet flowered pea plants and crossed them with homozygous white flowered pea plants. And in the F1 generation, he got all violet flowered plants. He then crossed individuals from the F1 generation and created an F2 generation. In the F2 generation, the white flowered plants reappeared. And what was interesting was that the purple flowered plants and the white flowered plants occurred in almost a perfect 3-to-1 ratio. So we had purple and white plants in the parental generation in a monohybrid or a true breeding cross, then had in the F1 generation, all violet flowers, and in the F2 generation, we have a mix of violet and white flowers. And from those simple experiments, Mendel was able to determine the way that simple characteristics and simple genes are inherited through dominant and recessive alleles. So again, a true breeding pea plant or a true breeding individual is an individual that is homozygous for its trait. In his experiments, Mendel chose plants that had purple flowers and bred them with other purple flowers until every time the purple flowers were bred, they always had purple offspring. And that was how he was able to determine and ensure that he had true breeding purple pea plants. And then he also would do the same thing for the white, which we know that the white is recessive, and so it's going to be quite a bit easier to ensure that the white color is true breeding. So in this situation, we end up with a monohybrid cross in the F1 generation. A monohybrid cross is where two heterozygotes are crossed with each other. And then in the next generation, the F2 generation, we had 75% purple and 25% white flowers. And this occurs because the parental generation involved a cross between true breeding violet and true breeding white flowers. All of their offspring were then heterozygous for this color. And then in the F2 generation, when some of those heterozygotes are bred together, they are then going to create both violet and white flowers. So that recessive trait, that white color, is going to show back up in the F2 generation after the parental generation's genes are no longer true breeding in the F1 generation. So if we have an individual who has the dominant phenotype, so the dominant physical characteristics, how can you tell if that organism is homozygous dominant or heterozygous in their genetics? To figure this out, we can do what's called a test cross. And so in that, you would take the individual in question and breed them with an individual known to be homozygous recessive for that trait, and then look at the offspring and determine whether that individual was heterozygous or homozygous dominant. So if a homozygous recessive individual is mated to a homozygous dominant individual, all of the offspring are going to be homozygous dominant phenotype because they're all going to be heterozygous genotype. On the other hand, if a heterozygous individual is bred with a homozygous recessive individual, half of the offspring are going to have the dominant phenotype and half of the offspring are going to have the recessive phenotype. In this case, round shape would be the dominant trait and wrinkled shape would be the recessive trait. In genetics, one of the laws of genetics is the law of independent assortment. This states that you assume that genes are inherited independently, that genes independently assort during meiosis. So we assume that any gene combination is possible in any offspring. This is generally true. However, if two genes are located close together on a chromosome, they're more likely to be inherited together than they are to be inherited separately. A good example of this is an individual who has red hair, possibly freckles, and doesn't tan very well. Generally, if you have red hair, you're going to have freckles and very fair skin. However, once in a while, you will have an individual who has red hair, no freckles, and can actually get a tan. But the alleles, the genes that code for that red hair color and freckles and fair skin are located very close together on a chromosome. And so most of the time during crossing over events and things like that, during meiosis, those genes are going to be linked together because they're located so close together on a chromosome. However, most genes in general are going to be able to independently assort from each other so that you have quite a big mix of genes and genetics in your next generation. And for our purposes, unless it's otherwise stated, you can assume that each gene is inherited independently of other genes. So when we're talking about multiple genes, or when we're talking about genetics, up to now we've talked about one gene at a time. In reality, individuals have thousands or tens of thousands of genes in their DNA. So when we're talking about something like pea plants, two of their main characteristics that we study often are shape and color. So an individual that is round would have a homozygous dominant or heterozygous genotype, and an individual that is wrinkled would have a homozygous recessive genotype. Color is also simple Mendelian inheritance, and yellow color is dominant, so it would either be homozygous dominant or heterozygous, and green color is recessive, so that would be a homozygous recessive individual. So if an individual is round and yellow, it's going to have the dominant genotype as well as the dominant phenotype. An individual who is wrinkled and green will have a homozygous recessive genotype for both of those characteristics. So in this case, a round yellow pea would have a full genotype, for example, of big A, big Y, and a pea that is wrinkled and green would have a genotype of little a, little y. A dihybrid cross is a cross between two individuals where they are heterozygous for two different genes. Both individuals are heterozygous for two different genes. In this case, we have big A, big Y, little I for both individuals that are breeding. This is a special case in genetics because it causes offspring that have an interesting ratio of phenotypes. When we're talking about color and shape in pea plants, the color and shape would occur in a 9:3 to 3:1 ratio of dominant and dominant, dominant recessive, recessive dominant, and recessive recessive. So let's look at how that can occur. So again, a dihybrid cross is a cross between two individuals that are both heterozygous for two different traits. And we're going to create from that the probability that we will have a 9:3 to 3:1 ratio of individuals who are dominant for both traits, dominant and recessive for the two traits, recessive and dominant, and recessive for both traits. And this is how we would set up a Punnett square for a situation where we're looking at two separate traits. So you have each possible combination of genes for both of the parents. So it's big A, big Y, big A, little y, big Y, and little a, little y for both parents and cross them. And it's color-coded here so that you can see where these different combinations of genes occur in this type of cross, where we have 9 individuals that are dominant for both traits, 3 individuals that are dominant for color and recessive for shape, 3 individuals that are recessive for color and dominant for shape, and 1 individual that is recessive for both of those genes. So when we're talking about inheritance, so far we've only discussed simple dominance, and that is probably the most common, but there are actually 3 different types of dominance and inheritance patterns when we're talking about genetics. So simple dominance is where you just have 2 phenotypes and 2 genotypes, which is the dominant and recessive. So dominant is, in this case, big B, or big B, little b. And then the recessive would be little b. Another possibility for dominance is called incomplete dominance. There are 3 possible phenotypes. So you have dominant, which is big B, heterozygous, which is big B, and recessive, which is little b. So in most introductory biology classes, the example that we give you for this is the example of flower color in snapdragons, where dominant, homozygous dominant genotype leads to a red flower color, a heterozygous genotype leads to a pink color, and the recessive homozygous recessive genotype leads to a white flower color. So instead of having 2 possible phenotypes, you can have 3, where the heterozygote has a different phenotype than either the homozygous dominant or homozygous recessive genotype. Another example of a type of dominance is called codominance. And this is where both phenotypes can simultaneously occur, such as in blood type. So in humans, our blood type is codominant, which is called ABO blood typing. Humans have each human has 2 alleles for blood type, but there are 3 possible alleles for blood type in people. So allele A would produce A antigens, allele B produces B antigens, and allele little I produces no antigens. The combination of these 3 genotypes creates 4 different potential phenotypes. So genotype big A would create type A blood. Genotype big A little I produces type A phenotype because the little I is the recessive trait in this. Genotype big B produces B blood type. Genotype big B little I produces B blood type. And this again occurs because the little I antigen, or the little I allele, is recessive to both the big A and big B allele. So A and B are codominant over little I. If you have little I as your genotype, you will have type O blood, which is the recessive genotype. The special case here is where you have an A allele and a B allele. In that case, you will actually have type AB blood. So in red blood cells, if you look down at the chart on the bottom, type A blood creates A antigens, type B blood produces B antigens. AB blood has both A and B antigens, and type O has no antigens. So this codominance in blood typing is important for individuals who need a blood transfusion or women who are pregnant, because if you try to mix B blood into A blood, you're going to end up having a reaction in that person's blood vessels where the blood will clot around those foreign antigens, which are the B antigens. People with type O blood are considered a universal donor because they have no antigens on them at all, and therefore O blood can be donated to any individual who is A, B, or AB. So O is the universal donor. Type AB blood is the universal recipient. So if you have type AB blood, you can receive blood from any person on the planet because your blood will accept A, B, AB, or O blood. If you have type O blood, you can only receive type O blood. Otherwise, your body will attack the incoming A or B antigens on the blood that is being donated. So another component of your blood type is whether you are positive or negative. So in my case, I'm O positive, which means that I have type O blood, and I'm positive for the rhesus or Rh factor in my blood. And so in blood type, that's important when you're receiving blood donations, as well as when a woman is pregnant and is having a child, especially if she has one after the first one. So if in this case, if we have a mom and a dad, the mom is negative for rhesus factor, so Rh negative, and dad is positive, so Rh positive. Dad's possible phenotype is either homozygous dominant, because being positive is the dominant trait. So dad is either homozygous dominant for that positivity of rhesus factor, or he's heterozygous. The mom in this scenario is homozygous recessive. And so when they have children, we would assume that 50% of their children would also be positive for rhesus factor, and 50% of their children would be negative for rhesus factor. And this is important because an Rh positive fetus can exchange blood with the mother. And so if the mother is Rh negative, it triggers the mother to make antibodies that attack Rh positive fetuses in future pregnancies. So there's a shot called a RhoGAM shot that will be given to the mother to keep the fetus safe so that the mother's body does not attack that fetus for having a different antigen in its blood than the mother does. Another type of potential inheritance is called polygenic inheritance, and this is when multiple genes contribute to a phenotype. So something like skin color or height are determined by an additive effect of polygenic inheritance, which just means that there are multiple genes that are acting together. So when we think about skin color, there's a range of skin colors in humans that ranges from very, very fair to very, very dark skin. And that skin color is determined by the amount of melanin that is produced by your skin. And it can range, in this example, we're assuming that it's affected by 3 genes instead of 2, and therefore you can either have a 0 on this scale, which would mean that you're very, very fair white, or you could be a 6 and inherit 6 melanin points, which means that you are very, very dark skinned. And so individuals that have a moderate skin tone are actually able to have offspring that have either darker or lighter skin because of that, because their combination of genes will have a broader range of possibilities of having either that dominant, which is the darker skin color, or the recessive, which is the light skin color. So in general, we think that that dark skin color is a dominant trait, light skin color is a recessive trait, but it's a combination of many genes that determine the color of your skin. So when we're talking about genetics, one of the things that people often are interested in is whether they are carriers of different types of diseases and traits. And so to determine whether a family or an individual or a couple has a possibility of passing on some kind of harmful trait to their offspring, genetic counselors can help diagnose genetic disorders in humans using a pedigree analysis. And this is just essentially where we take known characteristics or diseases in parents or grandparents or any level of a family, and then look at their offspring and through successive generations to see how that trait gets passed down. And so in simple terms, a female will always be a circle, a male will always be a square. And then if the individual is shaded in, it indicates that they are affected by that disease. We can also use pedigrees to determine whether a disease or a trait is dominant or recessive and whether it is autosomal or sex-linked. We'll talk a little bit more about sex-linked coming up, but for now we're just going to focus on autosomal and whether those traits are dominant or recessive. So a homozygous dominant individual has the disease or trait, heterozygous has the disease or trait, and homozygous recessive would have no disease or trait. So in this case, if it's a homozygous or an autosomal dominant disease, we would assume that it's going to occur in every single generation. Examples of autosomal dominant diseases or traits are polydactyly, which is where you have additional fingers or toes, and that is an autosomal dominant trait, or achondroplasia, which is where a person's limbs are shortened and the rest of their body is fully in proportion. And so if we look at a pedigree for either one of these diseases, or not diseases, either one of these traits, you will see that trait occur in every single generation. It's not going to skip generations, and individuals who have it, if they are dominant, are going to give it to every single one of their offspring. On the other hand, if a trait is autosomal recessive, you would assume that it's not going to show up in every single generation. If an individual is homozygous dominant, then that person does not have the disease or trait. If they're heterozygous, they don't have the disease or trait. They only have it if they are homozygous recessive for their genotype. Examples of this are albinism and cystic fibrosis. And for both of those, they are autosomal recessive. And so you would not assume that this would happen in every generation in a pedigree. You would assume that some individuals would be carriers, which are the ones that are shaded, just half shaded. And any individuals, and so this should skip generations because it should be a lot more rare than a dominant trait. So in humans, these are disorders that we know are controlled by a single gene. And so these are all autosomal disorders. So whether they're autosomal recessive, like albinism and cystic fibrosis, or autosomal dominant, like achondroplasia, Huntington's disease, and as well as others. So these are all autosomal, and next we're going to talk about sex-linked traits. So, in humans, sex is determined by by your sex chromosomes. All humans have 23 pairs of chromosomes. The first 22 pairs are your autosomes, and both men and women have those same chromosomes. And you have one pair of chromosomes that is your sex chromosomes. And men and women differ in the chromosomes that they have for sex chromosomes. Men are XY and women are XX. So sex-linked disorders are genetic disorders where the broken gene is on a sex chromosome. Most of the time, sex-linked disorders are going to occur on the X chromosome. It's quite a bit larger than the Y chromosome. It has quite a bit more genetic information, and therefore most sex-linked disorders are going to be on the X chromosome as opposed to the Y chromosome. And that has important effects for men in when we're talking about sex-linked disorders. So one example that we often give for an X-linked human disorder is color blindness. So there's a gene for producing color pigment that's used in vision, and it's found on the X chromosome. So your genotype is going to depend on both your sex chromosomes, whether you're a male or a female, as well as your allele for color blindness. So a female that is big A will have normal color vision. An individual that is little a will be a female with color blindness, and this is both with two X chromosomes. An individual that has two X chromosomes and is big A will be a carrier female, but will have normal color vision. On the other hand, males only have one X chromosome and one Y chromosome. So all it takes for a male to inherit an X-linked disorder is one bad copy of that gene. So in this case, we have a male who has an X chromosome with a little a allele and then a Y chromosome. And therefore that male will have color blindness because he has only that one allele that codes for color blindness. A male that has an X chromosome with a big A allele and then a Y chromosome will have normal vision. And so X-linked disorders are more common in males than they are in females because in females they have that extra X chromosome that can act as a barrier or a buffer to sort of protect that person from possibly having that disorder. They have two chances of having a good allele, whereas males only have one chance. So if we look down at the example on the bottom, we have a male, which is the XY on the left side of the Punnett square. The male has normal color vision because he has an X with a big A and then a Y. And the female has normal color vision because she's heterozygous, but she is a carrier because she has a big A and a little a on her X chromosomes. And from that, those individuals would have 100% of their daughters, the squares where there's two X chromosomes, all of their daughters would have normal color vision, and 50% of the sons that this couple has would have color blindness, and the other half would have normal color vision. So in males, X-linked disorders are inherited from the mother, not from the father. So male children receive their Y chromosome from their dad and their X chromosome from their mom. So any X-linked human disorders in a male are inherited from the mother only because the mother is always contributing an X chromosome to the child. A male will only contribute a Y chromosome. And so therefore in female children, an X-linked disorder could occur from either the mother or the father, but in a male child, an X-linked disorder is only inherited from that child's mother. And so that concludes our very quick review of genetics. So there we go.